right, welcome to this high-yield explainer where we are going to completely crush your microbiology board prep. Seriously, if you're a medical student staring down a massive, overwhelming syllabus right now, you are in the exact right place. Today, we're mapping out the founding fathers of the field, giving you the specific mnemonics, the tables, and the memory tricks you absolutely need to lock down those easy points for your exams. Here is our roadmap for today. We'll cover the early pioneers, then move to Louis Pasteur, the father. After that, it's Joseph Lister and antiseptic surgery, Robert Koch's discoveries, Koch's postulates and exceptions, and finally, Paul Ehrlich and his magic bullets. You know, it really helps to keep a mental timeline running in the background as we go. We're taking a journey straight from 1676, where we're merely seeing bacteria for the very first time, all the way to the mid-19th century, where we actually start targeting them with chemotherapy.
Okay, Section 1, Early Pioneers, the observers and the first vaccine.
Do not, I repeat, do not mix this up on test day. Antonie van Leeuwenhoek is your absolute patient zero of microbiology. Way back in 1676, he was the literal first scientist to see these invisible organisms. How'd he do it? By using the simple single-lens microscope that he built himself. And he famously called what he saw little animalcules. So if you see the phrases first to observe or single-lens microscope in a multiple-choice question, your brain needs to immediately lock onto Leeuwenhoek. Fast forward a bit to 1796, and we meet Edward Jenner. Now, what's really fascinating here is the etymology of a word we use every single day. Jenner developed the world's very first vaccine, and he did it to fight off smallpox. He actually used the cowpox virus to immunize children. Well, the scientific name for cowpox is variola vaccina. So the word vaccine itself, it's directly derived from his use of this cowpox virus. That is exactly the kind of high-yield trivia that examiners absolutely love. Section 2. Louis Pasteur, the father. The era of scientific discipline. So the crucial takeaway here is this. Louis Pasteur is officially the father of microbiology. Before Pasteur came along, people actually believed in spontaneous generation, or they thought that disease was just caused by bad air, miasma. Pasteur entirely disproved all of that. He postulated the germ theory of disease, firmly establishing that invisible microorganisms floating in the air are what actually cause disease. And that completely revolutionized medicine. Lock this acronym into your brain right now because it is pure gold for your exams. Remember VGPS to recall Pasteur's major contributions. V is for vaccines, and specifically, you need to know anthrax, fowl cholera, and rabies. G is for germ theory of disease, P is for pasteurization of milk and preservation, and S is for sterilization. He's the guy who developed the steam sterilizer, the hot air oven, and the autoclave. Also, make a mental note that he introduced the whole concept of liquid media using nutrient broth. VGPS. Burn that into your memory. Section 3. Joseph Lister. Antiseptic surgery. Killing germs on the outside. Think about it. Once Pasteur definitively proved that germs cause disease, well, someone had to figure out how to stop those germs in the operating room. Enter Joseph Lister in 1867. You need to intimately associate Lister with one specific chemical, and that's carbolic acid. He is the father of antiseptic surgery specifically because he observed that using diluted carbolic acid as a disinfectant to sterilize surgical instruments and literally clean out wounds massively reduced post-operative infections. So Lister equals carbolic acid equals antiseptic surgery. Section 4. Robert Koch. The discoveries. Isolating the culprits. Examiners absolutely love testing this direct comparison between Pasteur and Koch. Remember earlier we said Pasteur used liquid media? Well, Koch introduced solid media. He used agar as a solidifying agent, which, fun fact, was actually a brilliant suggestion from Ilchemius Hesse, the wife of one of his assistants. This massive switch to solid media allowed Koch to introduce methods for the actual isolation of bacteria in pure culture. It was a total game changer. Koch was an absolute master of identification. For your board exams, you must know his hit list. He discovered the specific bacilli for anthrax, tuberculosis, and cholera. He also gave us the hanging drop method, which is highly testable for identifying bacterial motility. Plus, he introduced staining techniques using aniline dyes. And whatever you do, don't forget Koch's phenomenon. This is the hypersensitivity reaction observed in guinea pigs that are already infected with tubercle bacilli when they get injected again. Section 5. Koch's postulates and exceptions. The rules of causation. So how do you definitively prove that a specific germ causes a specific disease? Koch laid out 4 really strict criteria to do just that.
Memorize the AIIR sequence for this. 1. Associate. The microorganism must be constantly associated with the disease lesions. 2. Isolate. You must isolate it and grow it in a pure culture. 3. Inoculate. You introduce it to a suitable lab animal, and it must cause the exact same disease. 4. Re-isolate. You have to extract the pure culture again from that experimental animal. Associate, isolate, inoculate, re-isolate. AIIR.
However, biology is messy, and let me tell you, examiners love exceptions even more than they love the rules. Use the mnemonic MTN to remember the organisms that completely fail Koch's postulates. M is for Mycobacterium leprae, and T is for Treponema pallidum. Why do they fail? Because they cannot be grown in vitro. They only grow in animals. And N is for Neisseria gonorrhoeae. This one fails because there's simply no animal model for it, even though it does grow in a petri dish. MTN is your fail-safe for these trick questions. Now, let's take a quick pause and look at this conceptually. By the late 1800s, thanks to Koch, we could find the germ. Thanks to Lister, we could kill the germ on our surgical tools. But here was the million-dollar question. How do we kill the germ once it is already inside the patient? Which brings us to Section 6. Paul Ehrlich, Magic Bullets, the Birth of Modern Treatment. Note this carefully for your exams. While Pasteur is the father of microbiology, Paul Ehrlich is the father of chemotherapy. He discovered an arsenical compound called salvarsan. This was the very first truly effective medicinal treatment for syphilis, and it literally initiated the concept of chemotherapy as we know it today. And Ehrlich wasn't just some one-hit wonder, he was an absolute medical powerhouse. He was actually the first to report the acid-fast nature of the tubercle bacillus. He developed tissue and blood cell staining techniques. He proposed the side chain theory for antibody production, along with the toxin-antitoxin interaction, which is actually known as the Ehrlich phenomenon. He even won a Nobel Prize in 1908 for immunology. And yes, the bacteria Ehrlichia was named directly in his honor. His ultimate vision was perfectly encapsulated in a single famous phrase, the magic bullet. He dreamed of a compound that could be shot into the body, completely ignoring the host's healthy cells and targeting only the pathogen. Salvarsan was the first realization of that dream. This concept perfectly bridges the gap from Leeuwenhoek's early microscopic observations straight into the era of targeted modern medicinal treatment. We've built an incredible foundation today, tracking the timeline of microbiology from the very first lens to the very first chemotherapy. Which leaves us with one final provocative question. Knowing this history and knowing how these brilliant pioneers shattered the limits of their time, who among you will discover the next magic bullet for tomorrow's untreatable drug-resistant diseases? Keep studying, keep questioning, and I will see you in the next explainer.